All set. Good morning, everyone, for joining our Grand Rounds series. And before introducing the speakers, few housekeeping items. As you know, this is broadcasting live, so anyone who's here, please use the microphone to ask questions. And you can view this via Vimeo and YouTube. The recording is also available for viewing after the Grand Rounds on our YouTube channel. And if you have any question, submit it, text DeBakey to 37607. Also, some of our upcoming training activities, we have an open aortic training on October 24th to 25th, CV nursing conferences on November 14th, and pumps and pipes on December 8th. So, well, where do I start? I have this distinct pleasure and honor to introduce someone I have admired a lot and thankfully have gotten the opportunity to work very closely, Dr. Svati Shah. She's the Ursula Geller Distinguished Professor of Medicine, Professor of Bioinformatics, and also Associate Dean of Genomics and Director of Precision Genomics Laboratory in the Duke School of Medicine, Vice Chief of Translational Research, and also runs an active clinical practice, Director of Adult Cardiovascular Genetic Clinic in the Division of Cardiology. Now, you know, we have a lot of folks who deal with very specific silos, population health, big studies, precision medicine, biomarkers. I've hardly seen somebody who's been able to put all the things together. And then again, having a conversation yesterday, what a diverse journey of training and something that our future trainees should look into, an MPH at Johns Hopkins, internal medicine at Brigham, cardiology training at Duke, followed by another master's in genetics. And he also played a major role in identifying very novel variants in the cardiometabolic pathways. But just beyond the traditional taking from bench to bedside, I think so she has played a major role in trying to close that loop from taking from bedside population back to the bench. So we are very grateful you being here, and we are looking forward in your thoughts beyond CAD in the heart failure omics, especially around the metabolism and beyond. So Swati, thank you so much for visiting. Appreciate it.
Thank you so much, Kurum. That was a really kind introduction, and hopefully I can keep you all engaged. There's some, I'm gonna warn you, there's a little basic science built into here, but I'll try to keep it relevant to patient care. So this is the most important slide that I'm gonna present today. If you pay attention to anything, pay attention to this. At this point in my career, this is what keeps us going. This is just a snapshot of the past couple of years of early career people that I've been able to work with across all different disciplines. And I think this slide also demonstrates the importance of team science. And if we really want to impact public health, we can't, as Kurum said, live in these silos. So these people represent clinicians, physician scientists, bioinformaticians, geneticists, graduates, PhD graduate students, masters in statistics students, all the way down to undergraduate students that we've had in the lab. So really, they're the ones who have done all the work. I sit on my, you know what, and try to get funding, and feels like that's all I do these days. These are the amazing people who have done all the work that I'll present today. All right, so I think everybody knows what omics is, but sometimes I'm surprised that people don't understand what omics is. So let me just talk you through this slide very quickly. So as Dr. Nasir said, I am trained in genetic epidemiology. I did a postdoctoral fellowship in genetic epidemiology over 20 years ago when the genome had not been sequenced. We did not understand variation in the human genome. And we knew with definity that cardiovascular disease was hereditary, right? Everybody knew that. If you had a parent who died of a heart attack before the age of 50, you knew that your risk was going to be higher. And so we were, there was a lot of hubris and a lot of excitement, you know, kind of when I started my fellowship and my postdoc in the early 2000s, that genetics was going to solve everything. So on the left-hand side of this omics is really the genome. It's the static DNA variation that all of us are born with. it turns out you don't die with the same variation that you're born with, which is an evolving field that I won't talk about today, but I'm happy to talk about if anybody's interested. And that was really cool. So then we sequenced, we, not meaning me, we sequenced collectively the human genome. And we did not figure out the genetic basis of cardiovascular disease. We put a lot of money and a lot of time and a lot of papers in my first R01, and we found lots of different genes, but we didn't really define the full genetic architecture of any kind of cardiovascular disease in the early 2000s. So at that point, for me in my career, there was a lot of serendipity in what happened. I had identified a gene that was a metabolic gene. It was called neuropeptide Y. It predisposed to early onset coronary disease, and I had found a promoter variant in that gene, and I wanted to understand if that promoter gene, back then we didn't have big databases that we could look at this online, right? So I had to take this gene and I had to try to figure out, does it actually have a functional effect? What does that mean? Does it actually change the relevant protein, right? So if you have the A allele versus the G allele. It's a metabolic protein. I went to one of our PhD biochemists and I said, can you measure this protein? A very simple question. And so he and I ended up forming a really great collaborative bond. And he had started developing, this is again in the sort of, now it's around 2005, 2006, had developed a really cool mass spectrometry-based platform for very accurately measuring metabolites, okay? And so we formed a collaboration in this very kind of hypothesis-focused space around this gene that I had discovered, but it opened up this whole space of being able to use metabolomics, measuring these little tiny things that are in nanomolar levels in your blood, which are the byproducts of cellular processes. So omics is really to say, look, static DNA variation is really important, right? That's the reason we all look the way we do and a lot of our health and disease. But it's how that static DNA variation dynamically changes over time, how it's transcribed, how that transcription then leads to changes in protein levels, how those proteins, as they undergo cellular processes, then result in these very small metabolic byproducts. And importantly, how that all interacts with the environment and the background of your genetics to then produce the phenotype. We were talking about the exposome last night, and I think there is a huge gap in when we talk about why did this person get disease and this person looks just like this person and they didn't get the disease, right? So all this stuff sort of together, and I think that's where omics in general, applied to human cohorts, can be a very powerful tool for trying to fill some of those gaps that we don't measure well in our patients. And really, I think, not to offend any of the basic scientists in the room, I think omics has really turned discovery science on its head. So this was something Rob Gersten and I published last year in Circulation as part of the American Heart Association 100th Year Centennial Series, where we used to rely on the mouse, right? And you knock out the gene in a mouse and you figure out what the phenotype is and then you can take that into the human, right? And then people talk about this valley of death of discovery, the time it takes to go from a mouse to something that actually goes to a human. But we have really turned this discovery science onto its head. You can start with this omics. So this is the static DNA variation, which is really important, but then how it gets transcribed into RNA, into proteins, into metabolites. Importantly, we talked about this last night at dinner, the phenotype, the disease, how well we understand the patient is really important, but we can start with human beings and identify targets that are relevant to cardiovascular disease. We don't want to ignore the mechanistic work, right? But you can start with the human cohorts using omics, identify signals. It's really important, that team science is really important, the retro-translation, where you find something in a human, you hand it off to the basic scientists, you let them mechanistically investigate it, and then there's that cycle of discovery where there's forward translation. They then look deeper in the preclinical model. I think this is going to exponentiate over the next 5 years. We have huge publicly available databases that you can get onto on your computer right now and identify drug targets for a disease that you're interested in. You do it, and actually probably be more interesting than listening to my talk. You just get on your computer right now and find some of these databases. So I think discovery science has turned on its head. So with all that, let's talk a little bit about heart failure. I won't talk about the clinical parts of heart failure because I'm actually not a heart failure doctor, and you all know more than I do. But I would say in heart failure, we need better biomarkers and we need better biology. Many millions of patients in America living with heart failure, with actually over 50% being HFpEF, and by 2030, heart failure will affect over 3% of the population. In heart failure, we actually have pretty good biomarkers. When you take into account like how few biomarkers we use clinically, there are actually some pretty good ones for heart failure. NT-proBNP, obviously, galectin-3, soluble ST2. We use high sensitivity troponin, GDF-15, a really cool sort of emerging biomarker. But I would still argue we need better biomarkers. Why do we need better biomarkers? I think we need better biomarkers because we need to identify heart failure subtypes, right? Not all patients with heart failure are the same. They should not all be treated the same. Even the HFpEF, HFrEF distinction, right? In a lot of ways, there's a lot of overlap, and within each of those diseases, there's a lot of subphenotypes. We also need to better understand the biology of heart failure, also to understand those subtypes. I think one of the biggest sort of potential clinical uses is in response to interventions. How do we determine what type of intervention a given patient with heart failure is going to respond to, and then how do we monitor response to that intervention? And then finally, identification of new therapeutic targets. This always feels like a holy grail, and people talk about the 17 years of the valley of death of you do something, you discover some kind of molecular target, you develop a drug for it. This is not 17 years anymore. So I think you're going to see a lot of new medications that are coming out over the next 5 to 10 years, including gene therapies. Okay, so what am I going to talk about today? This is an outline. I'm going to review a little bit about cardiac fuel substrate utilization. I find this really fascinating. To me, like how the heart uses fuel is really interesting, and we've done a lot of work in understanding the biomarkers and biology of cardiac fuel substrate utilization. In the end, who cares if you can't then take it to a patient? So we'll talk about some metabolic therapies in heart failure and how we're targeting metabolism in heart failure. I mean, we, the scientific community. Talk a little bit about work that Dr. Nasir and I are doing and how cardiac fat is actually a metabolic organ, and then briefly touch on heart failure genetics, which is really my old baby, which has become a new baby, renewed now that I think about clinical impact. Okay, cardiac metabolism. I promise this won't get too deeply basic. The heart has incredibly high energy demands. Both of my middle school kids know that the mitochondria and cardiomyocytes occupy greater than 1/3 of the cytoplasmic volume, which is pretty incredible, right? They're just the powerhouses. Cardiomyocytes are the powerhouses of the heart. But the heart has lots of different choices for fuels that it will use so that it can really make this incredibly high energy demand. So I've sort of demonstrated this as a hangry doctor, and I don't know what your cafeteria looks like. We're in North Carolina. There's not such healthy choices. So this hangry doctor is at the cafeteria, and they're trying to decide what they should eat. And there's all these different choices, right? Should they get a protein load at Chick-fil-A? That always has the longest line, so it might take longer to get your protein load. Should you just go to the salad place? That always has the shortest line in North Carolina. But, it's the healthiest choice. But what happens if the salad place is closed and you have to get the protein load? So in the same way, the heart has lots of different fuel choices. It can use fatty acids. That's sort of the salad, fish place. It can go, we do a lot of Krispy Kreme. Do you guys have Krispy Kreme here? Okay. Well, we do a lot of Krispy Kreme in North Carolina. Maybe it wants, glucose. There are also alternate fuel substrates, ketones. We're going to talk a lot about ketogenesis, and branched chain amino acids at Chick-fil-A. And really, this is super important that the heart can use all these different fuels to generate energy, because cardiac output is a high energy demanding process. 10 to 20% of the energy that your body uses every single day is just for cardiac output. So this metabolic flexibility, the ability for the heart to use different fuels, is central to myocellular homeostasis, to the healthy heart, the ability to use all these different fuels. So with that being said, in the normal resting heart, the heart primarily relies on fatty acids and fatty acid oxidation. As your heart starts to fail, or if you're exercising or it's under stress, it's not that it stopped using fatty acids, but it starts to preferentially depend on glycolysis and use of glucose. And this is beneficial in the short term, right? It's sort of like, oh, I can get a lot of energy from this glucose, but it's actually maladaptive in the long term. It's not an efficient ATP generation process. So over a long time of being reliant on glucose, the failing heart becomes metabolically inflexible. And I told you metabolic flexibility is a hallmark of myocellular homeostasis. So as the heart starts to fail, it's a vicious cycle of using glucose and becoming more and more metabolically inflexible with this very inefficient ATP generating fuel. We have learned over the past few years, and this is evolving science that has a lot of controversy, that there may be alternate fuels that the failing heart can use, some of which may be better even than fatty acids. And so we're going to talk about some of these today. So ketones being one of these alternate fuels, and branched-chain amino acids. My lab has studied branched-chain amino acids for 15 years, and I'm actually not going to talk about those today. I'm really going to focus on these fatty acid oxidation pathways and sort of a really interesting scientific and potentially therapeutic story around ketones. Okay, so fatty acid oxidation. I'm going to tell you a little bit of a story around plasma long-chain acyl-carnitine metabolites. So I had a really talented, actually he was a medical student at the time. He's a cardiology fellow now at UT Southwestern, Wynn Hunter. And Wynn asked a very simple question at the time. We had this really cool metabolomics platform with these highly accurate, targeted metabolites that we could measure in the blood that were focused on these fuel substrate metabolites, and we could pick up metabolites in the blood that were present at nanomolar level. When we first started doing this work, Rob Califf, when he was trying to describe metabolomics to people, he would say, it's like looking at somebody's trash the morning after to see how the party went the night before, right? That like you're looking at the byproducts of, either healthy or adverse cellular processes. So he asked a simple question. He said, like, can we identify metabolites that are associated with HFpEF? So we looked at this large number of metabolites, and for those of you who do computational work, this is a big data problem, right? You have sometimes hundreds, in this case hundreds, sometimes thousands, sometimes millions of things that you're measuring in the blood, and you're only looking at a much fewer number of people, right? Frank Harrell used to say you should have at least 10 people for every one variable that you're looking at, and we're flipping this multifold. So we use a lot of dimensionality reduction approaches, right? These metabolites are correlated, they live in the same biologic pathway, so we use machine learning approaches on the data to reduce the dimensionality. So in this case, we use principal components analysis to cluster these metabolites. And then we said, by the way, these clusters really reflect, interestingly, those mitochondrial processes around how the heart uses fuels. So using principal components analysis, we found all these clusters, and Wynn said, which of these clusters is associated with HFpEF? We have a large biorepository. You guys have a lot of resources here, I learned, around doing biobanking. We have 10,000 patients that we've collected in the cath lab. I can say we in that case because I actually was the fellow who consented for that, so I can take some ownership of this biorepository called CathGen. By the way, all these data sets I'm happy to collaborate with people on. Kuram and I have been doing a lot of collaborations, just submitted an R01 together last week, so happy to share anything and work with people. So what we found was there was this cluster of metabolites that even my biochemist colleague didn't really understand very well, and they were long-chain acyl-carnitines. And he found that these long-chain acyl-carnitines were associated with HFpEF. And then when we looked at these clusters, it was a bunch of different of these long-chain acyl-carnitines that were highly associated with HFpEF. But interestingly, we had two sets of controls. We had no heart failure controls, and we had HFrEF, heart failure with reduced ejection fraction controls. And what we found is these long-chain acyl-carnitines actually showed a linear relationship when we went from HFrEF to HFpEF to no heart failure. Yes, they were associated with HFpEF, but it was because they were intermediate in this sort of relationship between HFrEF, where they were the highest, medium levels in HFpEF, and then lowest levels in no heart failure. And we talk a lot about the molecular differences between HFpEF and HFrEF, and there's a lot of pathways that are very different between HFrEF and HFpEF. But really the lesson here is when we talk about fundamental canonical fuel substrate metabolism, the heart does not deviate, right? These are fundamental pathways. And in that sense, HFpEF is on the spectrum of HFrEF and no heart failure. And in fact, when we looked at ejection fraction, there was a linear relationship between circulating levels of these metabolites with ejection fraction. And then we looked in a data set. This is Mike Corey. He's a cardio-oncologist at Duke, and working with him and Lee Jones, we looked at a data set of women who had been treated with cardiotoxic drugs for breast cancer, but their ejection fractions were normal, so they were done with their therapies. And we actually found that these long-chain acyl-carnitine metabolites correlated with global longitudinal strain. So normal ejection fraction, but subclinical measures, right, of myocardial dysfunction when you have abnormal strain, and these metabolites correlated with that. So the message here, again, these canonical metabolism pathways are common between HFpEF and HFrEF. There's just varying degrees of disturbance. And then that's number one. And then number two, these metabolites may be markers of subclinical myocardial dysfunction, which, again, when we think about biomarkers, that might be a really useful biomarker. So then we asked the question, but are these prognostic? So those were all correlative, right? Those are all cross-sectional analyses that we were doing. So then we said, but do these actually predict what somebody's outcomes are? This is a very, very talented, now junior faculty at UT Southwestern. She was a cardiology fellow who did a couple of years in my lab. She's a heart failure transplant cardiologist named Lauren Truby. She is setting the world on fire. You guys should keep an eye out for her. I also sat with a lot of people at dinner last night who were setting the world on fire. So again, this is what really keeps me motivated. Lauren then asked the question, what metabolites predict incident MACE in patients with heart failure? So in this particular study, this was a nested biomarker study within the HF Action trial, which randomized patients with HFrEF, heart failure with reduced ejection fraction, to exercise versus no exercise. I'm really lucky. We have a big clinical trial institute at Duke called the Duke Clinical Research Institute. So I get to do a lot of biomarker and genetics discovery nested within those clinical trials. So you'll see a lot of our work here where we're doing a lot of work where we're the ones leading kind of the biomarker studies. So she asked, again, we took this set of metabolites. We did a lot of multidimensional data reduction to identify clusters of metabolites. And our hypothesis was around these branched chain amino acids, because that's what my lab has been studying for 10, 15 years. But in fact, what we found, it was, again, these long-chain acylcarnitines, circulating nanomolar levels of these long-chain acylcarnitines that predicted MACE in patients with HFrEF. In the HF Action trial, patients with diabetes responded less well to exercise. Okay, so exercise was overall beneficial in HFrEF, but not so much in diabetics. So we also looked at interactions with diabetes, and in fact, so the orange lines are the patients who had diabetes, and the blue lines are the patients who didn't have diabetes. This is a Kaplan-Meier curve, and you can see that it was really the patients with diabetes that these long-chain acylcarnitines seem to predict events even more so than in patients without diabetes. And there's a whole metabolic pathway of fatty acid metabolism in patients with diabetes that I'm not going to talk about today that could explain some of these things. And by the way, your levels of long-chain acylcarnitines in these patients who had HFrEF predicted what your response to exercise was going to be, right? So it suggests that we can take this little tiny snapshot and we can understand a patient with heart failures sort of very granular biology that we can't pick up very well clinically, and it might prognosticate not only what's going to happen to them, but perhaps even more importantly, might be able to predict how they respond to an intervention, in this case, exercise. We then wanted to understand how this played out in patients without heart failure. This is another person who is setting the world on fire. I was lucky enough, she decided to stay at Duke, so she's a first-year cardiology faculty member. Jess Regan, she is a preventive cardiologist. She is really interested in how your DNA changes over a lifetime. So in case you didn't know this, the DNA that you die with is not the DNA that you're born with. And you guys probably have heard these stories about the twin astronauts, identical twins, same DNA that they're born with. One goes into space, one doesn't go into space. The guy who went into space comes back and his DNA is different than his brother. That's what she's studying. So she's studying somatic mutations that develop over time. So in addition, Jess is interested in metabolism and preventive cardiology. So she asked, again, lucky to get to nest this within these clinical trials, asked the question of, in patients with diabetes who don't have heart failure, what metabolites predict incident heart failure and adverse events? And again, we found these long-chain acylcarnitines and medium-chain acylcarnitines, all of which are in this fatty acid oxidation pathway, that were the most predictive of incident heart failure events in patients who have diabetes. This is the data problem that we're dealing with. That's why this heat map is here, and then here are the specific metabolites. And Jess has done an incredible job with sort of understanding these metabolic pathways in heart failure. Okay, to finish up the story around fatty acid oxidation, what are they? They are markers of impaired mitochondrial fatty acid oxidation. I'm not going to go through this whole pathway, but basically you have these long-chain fatty acids. These are, partially from your diet. These long-chain fatty acids have to become energy, right? I told you in the normal fasting, resting heart, you're really dependent on fatty acids. And even when you're in heart failure, you still need those fatty acids. Those fatty acids get transported across the outer mitochondrial membrane by CPT1, and they become acetylcarnitines, and then get transported across the inner mitochondrial membrane and become these acylcarnitines. These acylcarnitines need to get shuttled into the TCA cycle for energy. When they accumulate, so when we find higher levels of these long-chain acylcarnitines in the blood, when they're accumulating, that means there's a bottleneck, right? They're not getting fed into the TCA cycle for energy. Why is that bad? Well, it turns out when these long-chain acylcarnitines spill out from the cell into the circulation, they can have pleiotropic, these are all preclinical studies that have been done, pleiotropic badness, right? Electrophysiologic disturbances, inflammation, cellular stress. We talked about metabolic inflexibility. So not only are these markers of badness, they, based on preclinical data, actually cause the badness. They are actually toxic in and of themselves. And then finally, we talked a little bit about this last night at dinner. This is always a problem in cardiovascular. I mean, people always say, like, oh, why is oncology so far ahead of us? They have access to the tissue, right? And in general, people don't want to give us pieces of their heart, just to study. So we're always a little bit stuck in understanding when we find something in the circulation, where is it coming from? Is it actually coming from the heart? So this is a story that unfortunately hurt my heart because my hypothesis wasn't true, but it's really important to test hypotheses that end up not being true because that means you're asking questions that are worth asking. And we were asking a really simple question, which is, where are these metabolites coming from? We're finding higher levels in the circulation. We think it's from fatty acid oxidation bottlenecks. The heart is not able to use the fatty acids as well in heart failure. They must be coming from the heart. They're not, it turns out, not in majority. So what we did here is we took explanted hearts, explanted failing hearts. We also had donor hearts that hadn't been used. Now, are those normal? We can have a debate about it, but they weren't overtly failing. So we had non-failing and failing hearts. We stratified them by diabetes. These are long-chain acylcarnitine box plots, highest levels, and then we mushed up the heart and did metabolomics, bulk metabolomics. The highest levels are in the normal hearts that don't have diabetes, and then they get lower and lower in the combination of heart failure and diabetes. We have a very small study where we have paired samples where we mushed up skeletal muscle and did metabolomics and then looked in the circulation. There's a much higher correlation, positive correlation, between what's going on in your skeletal muscle and what we find in the periphery, in the circulation for metabolism, which, if you take a step back and think, well, like what muscle makes up most of your muscle bed in your body? It's actually your skeletal muscle. And we know skeletal muscle is really important in heart failure, in this very vicious circle, promoting heart failure, but also promoting, muscle dysfunction, which we won't talk about today. So In the end, these circulating levels of fatty acid oxidation bottleneck metabolites are biomarkers. They seem to predict response to interventions. They're partially coming from the heart, but they're probably mostly coming from skeletal muscle. Okay, I'm going to tell you a perhaps even more interesting story. I find all this stuff interesting. I'm sorry if you guys don't. I think it's just so cool. My kids think it's really cool that the heart can use all these different fuels and that it determines what your heart does. Okay, we're going to talk about ketones, which has a lot more relevance, I think, to clinical care. That fatty acid oxidation stuff, we'll talk a little bit about what its clinical implication may be, but I'm going to tell you about this story about ketone biology. Okay, so does everybody know about ketosis? Does anybody know how can you, somebody yell out, like how can you achieve some ketosis right now? Maybe some of you are ketotic, right? Well, there's food in the back, so you're probably not ketotic. Yeah, fasting. So this is like an emerging space. But where are these ketones coming from? So the ketones, you have these fatty acids, partially from diet, and then the fatty acids go into the liver mitochondria, and they generate these ketone bodies. These ketone bodies, like beta-hydroxybutyrate and acetoacetate, can then go to cholesterol synthesis, or importantly, which is what we're going to talk about today, these ketones can get shuttled to the mitochondria across the body. We're going to talk about the cardiac mitochondria. They get shuttled to the cardiac mitochondria and can be used for fuel. So I told you that failing heart is super reliant on glucose. Well, maybe these ketones are an alternate fuel substrate that we're just beginning to understand. And people think these ketones are really cool and very interesting because they actually may be more energetically efficient. They may be better fuels for the heart. They actually generate more ATP per unit of oxygen than fatty acids and glucose do. So they may be more efficient fuels, and they go through separate pathways. Glucose and fatty acid utilization are highly linked to each other, right? One goes up, one goes down. Ketone catabolism is a different pathway. So it's sort of independent of that, and sort of this is an evolving biology around the heart's use of ketones as an alternative fuel source. And in fact, when you have low levels of ketosis, like when y'all woke up this morning, this low levels of ketosis in humans has been shown to have pleiotropic beneficial effects. Improvements in endothelial function, oxidative stress markers, mitochondrial function, including in the skeletal muscle, decreased inflammation, improved cardiac remodeling when we look at echo parameters, blood pressure, body weight, cardiac energetics, again, when you do CPXs and echoes on patients who have low levels of ketosis. This is a really cool paper that's not from us. This was published by Danielle Murashige, who's an MD PhD student who's actually applying for cardiology fellowships now. She did a really cool study where she took patients who were undergoing EP procedures, and so they were already getting BiV pacemakers, right? So they were getting a lead put into their coronary sinus. So what the electrophysiologist did is they stuck a catheter into the coronary sinus, which they would do for the pacemaker lead, and they took blood out of the coronary sinus. So they had coronary sinus blood, arterial blood, and venous blood. So they could calculate how much stuff the heart's using, and then how much stuff the leg is using by comparing the arteriovenous and coronary sinus gradient. I'm going to tell you about another junior faculty who's doing similar work here at Duke in my lab. So doing that, she basically looked at a bunch of different metabolites and found that in the failing heart, this is HFpEF and this is HFrEF, there is increased ketone utilization. So 2% baseline with no heart failure, 6.4% with HFpEF, 16% in HFrEF. So that hungry heart that is reliant on glucose is also desperately potentially looking at these alternative fuel sources, including ketones, which again may be more efficient fuels. I'm going to tell you a little bit about Senthil. Again, the most important part of my talk are these junior people. Senthil Selvaraj is really talented junior faculty who I'd say is one of the world's leaders in thinking about ketone biology. And he is currently a junior faculty in my lab, but very independent and doing really cool work. This is work that he did where he showed that circulating levels of ketones, so as biomarkers are really important. They are associated with all kinds of different, we did what's called a phenome-wide association study looking at all different kinds of disease phenotypes, and these circulating ketone levels are associated with heart failure, right ventricular systolic pressure, a lot of cardiac function parameters, and predict incident heart failure. All right. why do we care about any of this stuff? Like, okay, we talked a lot about this, like who cares about biomarkers, right? I'm gonna tell you a little bit about, I think you're gonna see over the next few years, more therapies that are metabolic therapies for heart failure. I hope that all of you are thinking in your head right now, we already do that. Y'all are using SGLT2 inhibitors in your patients, you're using GLP-1 receptor agonists in your patients. Metabolic modulation of patients with heart failure, right? It is happening in 2025, and there's potentially new stuff coming down the line that will be metabolic modulators that may help our heart failure patients even more. So I'm gonna tie some of these therapies to this sort of basic science biochemistry that I told you about. So the first thing, I told you about those long-chain acylcarnitines, which are markers of this impaired fatty acid oxidation. So a question is, can you modify them? This is work that was, on the left is work that Tariq Ahmad did when he was at Duke with us, and then on the right is work that was done by Robert Gersten's group, not by us, where we found that circulating levels of these fatty acid oxidation long-chain acylcarnitines are modifiable in heart failure with mechanical offloading. So in this case, in the study that Tariq Ahmad did at Duke, we took patients who had chronic heart failure, end-stage heart failure, who were about to get an LVAD, and then we measured the same metabolites after they got their LVAD. And we found that these metabolites, these long-chain acylcarnitines, decreased after the patient got the LVAD and returned to sort of chronic heart failure levels, so mechanical offloading. Similarly, Robert Gersten did a cool study where he took patients who were undergoing TAVR and did pre and post metabolomics and found that these long-chain acylcarnitines, once you relieve the stenosis, decrease. So mechanical offloading can modify these metabolites. There's a bunch of other fatty acid oxidation modulators that are in various stages of being tested in heart failure. It's interesting because it turns out some of these decrease fatty acid oxidation and some of them increase fatty acid oxidation. Decreasing fatty oxidation may be a good thing because maybe it can use glucose and fatty acids, glucose and ketones and other fuels better. Increasing fatty acid oxidation could be good because it relieves that bottleneck and allows those acylcarnitines to be used for ATP generation. These three top drugs decrease fatty acid oxidation. A lot of them have had hepatotoxicity issues, so I don't think they'll come kind of into clinical play. This drug called TMZ has shown a lot of promise in patients who don't have heart failure and patients with diabetes, and there's some early data around this drug in heart failure. There's also data on increasing fatty acid oxidation. These ACC2 inhibitors are showing some promise. PPAR alpha agonists and fibrates have been around for a long time. They show benefit in patients without heart failure and are starting to be tested more in heart failure. But I think the most important and cool thing is these drugs that aren't specifically for fatty acid oxidation inhibition, but are used for metabolic treatment and also have effects on fatty acid oxidation. So here's a study that we did, again, led by Jess Regan, where we looked at these metabolites with GLP-1 receptor agonists. And we found that GLP-1 receptor agonists beneficially modify these fatty acid oxidation metabolites, and it's one of the main pathways metabolically that are modified. So GLP-1s seem to improve fatty acid oxidation and seem to be one of the reasons that mediate the beneficial effects that we see with GLP-1s. I'm going to talk to you a little bit more about SGLT2 inhibitors in a study that we did, which also modify beneficially these fatty acid oxidation pathways. And then last year, I think this drug is interesting. We'll see how it plays out. It's called HU6 because it's still kind of, just in phase 2. It was presented last year at HFSA, the Humane trial by, I think, Delane Kitzman group led it, specifically focused on obesity HFpEF, so patients with HFpEF who have obesity-mediated HFpEF. And they use this HU6 drug. This HU6 drug actually hits multiple pathways. So it relieves fatty acid oxidation bottlenecks, but it also relieves glycolysis and improves glucose utilization. So it's a metabolic uncoupler, where now glucose utilization and fatty acid oxidation are not coupled together and relieve stress in both those ways. So this HU6 drug you may be seeing more studies on as a metabolic modulator specifically for HFpEF. So, okay, so we talked about, you guys said that you were fasting last night. You guys are all mildly ketotic when you got here today. Well, what about exogenous ketosis? We give you ketones. You don't have to fast, because I don't like fasting. So what about exogenous ketosis? So you can induce ketosis by endogenous ketosis, you're fasting or whatever, or you can induce ketosis exogenously. And so it turns out you can do this intravenously, not very convenient for our patients, but you can actually drink ketones. And I'm not going to tell you the brand so I don't look like a billboard, but you can Google it, and there are drinks that you can buy right now. They are not cheap. I've drank them. They do not taste good. Has anybody ever had a ketone ester drink? They don't, in my opinion, they don't taste great. So some of the studies that we've been doing that I'm going to tell you about, we let them pick the Crystal Light flavor or something to put into it because it doesn't taste awesome. But you can actually give people these ketone ester drinks. So you can give exogenous ketones. And by the way, how much your circulating level of ketones is directly determines how much ketones your heart uses. So the more ketones you have in your blood, the more your heart's going to use ketones. And I told you it might be an energetically beneficial thing. So this is a paper that came out last year in Circulation showing benefit of drinking these ketones in patients who have HFrEF. So it's a very small study. They took 24 patients. They gave them this ketone ester drink for 2 weeks. And in 2 weeks, I think it's pretty incredible. I'm not trying to be a billboard for ketone drinks, because these ketone drinks are also advertised for like improving exercise and improving your mental energy and a lot of stuff that's completely not tested. But in this very rigorous study, they found that, so in the orange are the patients who got the ketone ester drink, and then in the blue, people got isocaloric non-ketone ester drink, so basically placebo. And no surprise, your circulating level of ketones increased with drinking this drink. But look at this. Look at these differences in left ventricular ejection fraction, global longitudinal strain, more negative being good, E to E prime, and LA reservoir strain. So a lot of hemodynamic parameters that improved in these patients with HFrEF with just 2 weeks of home dosing of a ketone ester drink. This is work that we've done in our lab, again, with Senthil. My intervention was to put the Crystal Light in the ketone drink because I can't stand the taste of this stuff. So we took patients, again, with HFrEF, but we only gave them 1 dose of a ketone ester. We wanted to understand the safety and feasibility of giving a single dose, and we wanted to look at 2 different doses. So we used 250 milligrams per kilogram and 500 milligrams per kilogram. We found that the single dose was very well tolerated, had a very good safety profile. We measured blood at multiple time points up to 6 hours after the single dose of ketones. And we basically, we found mild improvements in cardiac energetics, but really what we found was really pleiotropic effects across all these metabolites that we measured. And specifically, these medium and long-chain acylcarnitines decreased pretty significantly, some of them almost 30-40%, with the single dose of ketones, suggesting that this, again, if you have ketones available, you are relieving this stress and fatty acid oxidation in patients with heart failure. Okay, so that was exogenous ketones. You guys fasted and got endogenous ketosis. There's another way to do endogenous ketosis that all of you use in your patients all the time, and that's SGLT2 inhibitors. So this was, what was thought to be one of the beneficial effects of SGLT2 inhibitors is it induces endogenous ketosis. Your liver makes more ketones. We know that these are foundational therapies in HFrEF and HFpEF, DEFINE-HF and PRESERVED-HF being 2 clinical trials that showed that SGLT2 inhibitors are not only useful in patients with diabetes and heart failure, but in patients who don't have diabetes and who have heart failure across the heart failure spectrum. So again, Senthil and I did Senthil's really just a powerhouse. We again did a nested sub-study, a biomarker sub-study within these 2 clinical trials looking at metabolites with the goal of understanding, well, is it really ketosis or is it something else that might be why these SGLT2 inhibitors are beneficial in heart failure? So what we found was what metabolites changed. We did find that these ketones changed. we would expect these ketones to change. The ketones increase slightly, very low levels of ketosis that were induced with SGLT2 inhibitors, and this was in patients with HFrEF. This was in the DEFINE study. But we also found a set of these acylcarnitines, which report on fatty acid oxidation, and we found that these acylcarnitines increased. These are earlier in the fatty acid oxidation process than these long-chain acylcarnitines, and we did not have an increase in the long-chain acylcarnitine. So it actually suggests that the SGLT2 inhibitors are increasing fatty acid oxidation also. So in addition to inducing ketosis, very mild ketosis, they're also relieving this bottleneck in fatty acid oxidation, which hadn't been shown before, the sort of pleiotropic metabolic effects of these SGLT2 inhibitors at the level of fuel substrate utilization. Then we looked in HFpEF, so we looked in the PRESERVE trial, no difference in any of the metabolites. SGLT2 inhibitors actually didn't even induce ketosis in HFpEF. And we were a little bit surprised by this. EMPA-Vision came out, showed the same thing, which reassured us that we had found something that was real. So then we combined both these studies, looked across the LVEF spectrum, and what we found was that these increases in ketones are really only seen with lower LV ejection fraction and may be more relevant in patients with HFrEF. But these changes in fatty acid oxidation metabolism, these medium and long chain acylcarnitines, so relieving that fatty acid oxidation bottleneck, were consistent across left ventricular ejection fraction. So actually these SGLT2 inhibitors, based on these data, may have their fatty acid oxidation effects may be more important in their beneficial effects in patients with heart failure, including patients with HFpEF, rather than the ketosis. And in fact, when we looked at actual intermediate endpoints in these trials, it was these long-chain acylcarnitines, these fatty acid oxidation markers, that correlated with clinical endpoints, changes in BNP, KCCQ. So it's not that the SGLT2 inhibitors don't induce ketosis, but it seems, based on these data, that it's actually their effects on fatty acid oxidation which is inducing these beneficial effects in heart failure. Okay, I'm going to finish up the metabolism stuff really quickly. We've done some work on GLP-1 receptor agonists, which I'm happy to tell you about, in more detail, in individual meetings. But we've done a lot of proteomics work, and we had an interesting conversation last night. Who cares, Darren McGuire, you were telling me, Darren McGuire says, who cares how the drug works as long as the drug works? Well, I happen to care how the drug works. So we've been doing a lot of work with GLP-1s. We have found that GLP-1s actually have quite pleiotropic effects. So when we look at pre and post, this is over 5,000 patients who got a GLP-1, and we measured levels before and after the GLP-1, and we found really marked pleiotropic effects. And these are just hot off the press data. GLP-1s actually slow organ-specific biologic aging, and particularly in the heart. So your heart's biologic aging can be slowed by GLP-1s. Can I have 5 more minutes? Jennifer's stalking me. 5 more minutes to talk? Okay. I'm going to tell you a really quick story that Khurram and I are working on. This is part of the R01 that we submitted last week. This is also a lot of hot off the press stuff. You guys have incredible expertise in cardiovascular imaging and a lot of really great research going on. This is work related to cardiovascular imaging using CT, where you can actually image cardiac fat. I think you all know this, right? And it turns out cardiac fat that actually opposes an organ. So we think about fat having endocrine effects, right? The fat in your liver secretes stuff, and that secreted stuff is really bad. The fat around organs has paracrine effects. So it secretes stuff that directly influences potentially cardiovascular disease. Epicardial fat actually shares a vascular supply with the myocardium. It shares vasovasorum with coronary arteries, for example. So that shared blood supply enables the cardiac fat to secrete bad stuff, which then goes into the coronary arteries or into the myocardium and can promote badness. And we can actually measure this cardiac fat on CT. You can measure the volume of fat, and you can measure inflammation in the fat. So we've been doing a lot of molecular work. These are studies that show that not only can you measure it, but it's important. In this particular case, they were looking at paracoronary inflammation, and it predicts incident events. And in this case, they were looking at epicardial fat inflammation measured on CT, which predicts incident heart failure. So hot off the presses, we have found several metabolites, but probably more importantly, we did a genome-wide association study in the PROMISE clinical trial. These are preliminary data for our R01. We actually found a single locus, a genetic locus, which is associated with heterogeneity of cardiac fat inflammation. And this particular locus mediates cytokine release in macrophages, the gene does. So a lot more work to be done in this space, but we've been linking a lot of circulating omics with radiomics. I'm gonna skip that. We're doing some single cell in fat. So I wanna finish up with just saying, don't forget the low-hanging fruit of monogenic causes of heart failure. So I have a renewed interest in genetics, even though I went off track with cardiac metabolism, because both of my kids were diagnosed with a genetic disorder a few years ago. They both have factor VII deficiency. And so I started thinking, like, there's so much, and I take care of these patients, there's so much undiagnosed genetic disease. This was work led by one of our cardiology fellows, Jawan Abdul Rahim. We took those 10,000 patients that I told you from the cath lab. So these are patients who are getting an invasive cardiovascular procedure, theoretically being seen by the best cardiologists at Duke in the world. And we did sequencing on them and then did phenotyping. And we found 2% of these patients not only carried an abnormal, a pathogenic mutation, they had evidence of that particular monogenic disease. But only a third of those patients had been diagnosed, right? So 2/3 of these patients with deadly monogenic, so this isn't just risk. It's not that just they carry the genetic disorder. They have evidence of the genetic disorder, and we are missing these diseases. So 10 years ago, my heart failure colleagues didn't refer anybody to me for genetic testing. And now there's a really renewed interest, peripartum cardiomyopathy, chemotherapy-induced cardiotoxicity. These patients should get, in my opinion, happy to have a discussion about it, but these patients should get genetic testing. That genetic testing needs to be done very carefully. So I'm gonna finish up with telling you very quickly about HeartShare. HeartShare is an NIH program on HFpEF. I co-chair the steering committee for this particular program with Javed Butler. I'm mentioning this to you because I think it's a really great resource for how we try to solve some of the problems in HFpEF. It has a large retrospective cohort where we've created a big database of like 30 different studies of HFpEF, and we're prospectively across 7 sites collecting new HFpEF patients. We have 750 HFpEF patients, 250 non-heart failure controls, and we're basically doing head-to-toe phenotyping, including imaging, RV biopsies in a subset, skeletal muscle biopsies, adipose tissue biopsies, doing single cell omics, circulating omics, cardiac CPXs, a huge amount of data. Please feel free to reach out to Javed Butler or I if you're in, and Sanjeev Shah is the head of the coordinating center. This is an ongoing study. We're about 600 patients into the 1,000. So with that, this is my many, many people that I get to work with, and I'll end with a quote from Arthur Ashe. These are really difficult times for all of you. I hope that I see a lot of early career people in the room. I hope that you will pursue a career in science. I know it's really hard, but even my husband's a full-time clinician. You can still build research into what you do. It's not easy, and this is something I tell myself in the mirror every morning. Arthur Ashe, a tennis player, unfortunately passed. Start where you are. Use what you have, do what you can. Start where you are means, okay, don't remind yourself of your failures. You are where you are, so start where you are today. Use what you have. What do you bring to the table? Each of you brings something really unique and really important to the table, so use what you have, and then do what you can. Every day, you just do what you can to make an impact on patient care and make an impact on what you can do for people around you. And then you sort of take each day one by one. So with that, I will end, and thank you so much for having me.
Well, before I take questions, what an inspiring talk, in the sense, giving us the entire 360 of what can be done. Clearly, we are all interested in the fact of why people have disease, why they don't, how they respond, why they don't respond. And I still think that what are the mechanisms are critically important. And I think we are in this era where we have an ability, as you pointed out, 20 years back, getting those 15 individuals, 20 individuals is hard. And we have, of course, national, international biobanks, but I think one of the most untapped space is within our own health systems. And clearly, the CathGen study, the effort that was put in. So the question I have to understand the entire Omics with the deep phenotyping, if in 2025, within the cardiovascular space, in the clinical settings, now with the EHR and the imaging, which domains would you recommend investing in creating those biobank infrastructure, especially in a place like Houston Methodist, where a lot of the cost and I would say logistics are being taken care. So what would you advise us on that?
Oh, wow. You're asking me to choose my favorite child, is what I'm hearing.
We would like everything, but if we have
know.
to start and create a structure and then scale it.
I think there are so many things that are unmeasured, right? Clinically unmeasured. And even so, I read echoes one day a week. I miss, there's so many things that you miss on echo, right? The human eye can only look at so many pixels of data and so many images. So I think imaging is a snapshot into what's going on in the human body and pairing that with AI. So making the investment, you guys have amazing people here who are doing this work, pairing AI with the imaging and our patients are getting clinical imaging. The imaging's already there, right? So using that imaging, taking a step back, the most important thing is actually the phenotype. So we talk about all this cool technology and what we can measure in the blood, and we're doing a lot of, I didn't show you the slide because I was going over, but we're collecting fat from people's hearts. Like in patients who are undergoing CABG, we're actually dissecting out that heart around the coronaries and the epicardium and doing single cell and doing all these new technologies. None of it matters if you don't understand the phenotype, if you don't understand the disease. And I think that's where imaging can be particularly helpful. And then can I have a second
Yes.
choice? The exposome. There are so many things that we take these little snapshots of our I think about my mother right now. My mother's going through a lot of medical stuff. What her doctors see when they spend 10 minutes with her in the clinic, but then what I see, it's so trite. I mean, all of us do this, but I'm really recognizing it in real time with my mom. And then I see what's happening at home. And she's got her bag of chips next to her, couch, right? And her doctors are not knowing that. She, whatever is happening in her house, and I see how poorly her house is put together. So I think the exposome and how we get better data, digital health is going to be a big component of how we can measure those things on patients outside of when they come to see us in clinic. So those would be my two. The exposome, I think, is more difficult than the imaging.
So what I'm hearing, at least more preciser phenotypes where for the same exposures or risk factors, you can see a spectrum of the disease that can at least give us some idea. And of course, if they're being treated, then we can also look at some of the response. But in fact, any of these questions, if we want to ask, let's assume for some of our young career folks in the next 3 years, it's going to be hard unless we have the infrastructure to provide those resources. Like we have 5,000 cases with CT angiograms with varying degrees of disease and progression. So I think so that's the investment that health systems and the future academic medical centers needs to go. And it's amazing to see this work that you and Javed and other are trying to give us a pathway through the NHLBI. Hopefully industry, health system, all of us can come together. So that's fantastic. Questions from, yes.
Well, I enjoyed your talk tremendously. This may seem like a strange question to come from a cardiac surgeon, but in cardiac surgery, we treat a lot of people with valvular heart disease and heart failure. And we've been doing this for about 75 years. And we measure gradients and we see our patients get better. But there's a lot more to the story. When we get down to the fundamentals, we're not really restoring normal function in a lot of these operations. And really the critical issue still hasn't been answered. And we even had a debate about this just a few weeks here about what is the minimum gradient relief in aortic stenosis you need to have a beneficial effect. And various techniques can produce anything ranging from a residual gradient of 10 millimeters to 30 or 40 millimeters. The patients all do very well. In the mitral valve field, we have a surgical technique we've developed here that completely restores all the normal hemodynamics of flow through the heart, through the mitral valve, and with preservation of vortex flow and all the rest of it. And loss of that produces about a 10% loss of function of the heart. There's a 10% energy deficit from that loss. So we don't lose that in our mitral repairs, but obviously a MitraClip, for example, does lose at least 10% of its energy. And we've been doing very well with our sick old patients because eventually they're going to die. But now we're rapidly moving into a younger population that's going to be exposed to these loads for years. And currently we don't have any good answer. And it just seemed to me sitting here that the answer may be right in front of us. And it may be one thing we could start looking at the levels of some of these critical metabolites in these different groups of patients who've had different types of procedures with a seemingly similar outcome that may in fact not have a satisfactory outcome metabolically.
That's a fantastic study design. Somebody here should do it. I'm going to tell my folks at
I was glad to
home.
see you've started with the TAVRs already.
Yeah, well, our guys are very collaborative with us, and here and at Duke, and our surgeons have been great about working with us on research studies. I think that's a fantastic study, pre and post MitraClip, and pre and post, some of these the surgical interventions, and understand what happens to these metabolites. We have data I should have shown it today. I didn't realize the surgeon was going to be in the room. We have data where we took transmatic hearts and actually did metabolomic profiling in the perfusate. And you can it's no surprise, right? As the heart becomes more ischemic, you see, increases in these fatty acid oxidation markers, decreases in branched chain amino acids and ketones as the heart starts to use it more. So I think you could very easily then transfer that signature that we know correlates with, adverse myocardial function, and translate it into which intervention is better as an intermediate, right? Because these young people, you'd have to follow them for a really long time to understand what the long-term effect. It's a great idea. If anybody here wants to do it, I'm happy to help you with the metabolomics. You'll do it? Okay, great.
Thank you, Conrad. So terrific lecture. So I have maybe one or two comments, and maybe for you to ponder on this. It's so intuitive that in patients that have heart dysfunction for whatever reason, whether it's pressure, volume, or chronic heart failure, that the heart may be malnourished. And the idea of doing metabolic adjustment to nutrition of the heart would be, it's an old idea. I mean, people have looked into many different ways. So I guess, why is it that we have not been able to find, other than maybe recently the SGLT2 inhibitors, maybe GLP-1 as you showed today, a better way to nourish the heart? I mean, what is it that's missing? Is it the fact that it's difficult to measure metabolites in a clinical setting? Is it the fact that we don't have the right therapeutic interventions? It's like, for quite some time, I've been thinking, why don't we just do a clinical trial on ketones in heart failure just to see outcomes rather than just short observational studies like that? So just your thoughts on that.
Yeah, I think those are excellent questions. I think one of the problems with a lot of these metabolic modulating drugs is the off-target effects, right? So that's been, I think, a limitation even in preclinical, all those CPT1 inhibitors that have been tried for a long time. So part of it is we can't target just the heart, right? I mean, with these, everything goes through the liver. So hepatic toxicity is a big issue with anything that you do that you're going to give orally or intravenously. It's gonna go through the liver if it's a metabolic modulator. So I think that's number one. Number two is, you already said it, we haven't been able to measure these metabolites. So those long chain acylcarnitines that I told you about, if you guys go look at some of these big population-based cohort studies, they are not statistically significant. And you say, well, how come they didn't play out in some of these big population-based cohorts? And you look, and it's because they didn't actually measure these. These have to be measured with, internal standards, targeted metabolomics. I think that is, a second big consideration. The third big thing with the ketones, I think, is that people were really dependent on, were using intravenous ketones for exogenous ketosis, right? And I think the development of some of these oral ketone esters is a relatively new thing. So there are now small, randomized trials that are showing benefit of the ketone ester drink. I showed you the HFrEF, but there's another study that showed that it is beneficial in HFpEF. These are very small studies.